receptors and signaling pathways involved in this specialized form of phagocytosis are known to quell inflammation. And so our thinking is if we can understand that process, we can maybe hijack that signal to quell inflammation in other disease models. Where Jedi comes into the picture is as one of these engulfing receptors that clears apoptotic cells. And our lab has shown with our preliminary results that Jedi knockout, or JKO, has been shown to decrease neurogenesis. With marker that works, and increase inflammation in these microglia. So the core question of my project is, how does Jedi regulate inflammation? To start getting at this project, my first aim here will be looking at directly how do we activate Jedi. So we know that Jedi is an engulfing receptor expressed on the surface of microglia. What our question is, what activates it? What signal in the apoptotic cells is its activating ligand? The first thing I'll be looking at is whether phosphatidylserine is involved in activating Jedi, since phosphatidylserine is a widely accepted eat me signal for apoptotic cells. It stands for that Jedi would be involved in recognizing phosphatidylserine to clear those apoptotic cells. We also think it's phosphatidylserine from homology studies where the Drosophila homologue of Jedi, Draper, is one of the first characterized glial engulfing receptors shown to clear apoptotic cells, specifically through recognizing phosphatidylserine. So aim one is going to be looking to try to identify ligand for Jedi 1. Our next step that we want to look at is what is the immediate downstream signal pathway of Jedi 1. So what mediators are going from the activation of Jedi to some sort of outcome on inflammation. And here we have a really interesting protein that associates with Jedi called psych, the spleen tyrosine kinase. So psych has been implicated by other groups, and there were 2 separate studies published in Cell 2022 that identified psych as a key mediator for microglia inflammation and metabolism. And so aim 2 will be looking to uncover whether that psych signaling is conserved in microglia. Lastly, we know that this spleen tyrosine kinase psych can trigger signaling cascades, as many kinases do. And so the pathway leading to some kind of nuclear change on some kind of nuclear output of inflammation-related genes, the sort of master regulator of inflammation with regards to nuclear transcription would be NF-kappa B. And so whether we see NF-kappa B activated or NF-kappa B affected as a result of Jedi activation will be what I'm looking at in game 3. Collectively, these works will parse out a signaling pathway from activation to transcriptional outcome that could provide numerous targets for potential therapeutic intervention when we're looking at regulating microglia inflammation. So,
started with the overarching question of why is this important? Why do we care? Why are we looking at it? Right? So giving some importance, and then we concluded with the impact of what the results would contribute to his field, and that would really tied it really nicely together. So that's something, if you want, you might consider doing.
Yeah, so I mean your introduction and ending, providing some of the technical structure stuff in the middle is where the audience would start to lose attention. That's probably where you want to pack that stuff in there. And then when thinking about the drawing, that was the hardest part for me coming up with this talk because I'm graphically inclined. And so my first trials of this had a lot of the points that I had here as full sentences pre-written out. And I worked on it with my friends, and they were like, that's too much, it's way confusing, then the people are worried about reading rather than what you're saying and talking about. So quick sketches, even bad sketches, are fine. And making it big and providing lots of space for them to then ask follow-up questions on is also a good approach.
I will say my drawings are also really bad. Like my PI really tapped that morning.
Yeah.
Like when I was practicing, and then when Bruce had me draw a neuron, and he was like, "Oh, that's new." So, and I still passed. So like you don't need to be an artist
Yeah, when I was drawing a synapse, made a poor mistake that resembled some other kind of shape that was not appropriate for a workplace. And so I quickly chuckled and moved past that, but that was a tough one to try to get back. Just make sure your synapse is flat. You can avoid that. Yeah, we got it. I don't know if questions and other stuff.
Yeah,
Or I can talk about some of the questions.
do you guys wanna talk, do you guys have questions, we can talk about this first, or you guys wanna move on to the volunteers who are drawing?
So if you don't have preliminary results,
Yeah, so preliminary results need just background information relevant to your project.
oh, okay, so it can be just
This
like
wasn't
I
stuff
didn't
that-
have prelim results for
Okay.
mine, like it was a new
Yeah.
project and it was just, I just gave background on Alzheimer's disease, vascular related, like dementia, and like how those correlated. But it wasn't necessarily like my prelim, it was just like background in research field, like why are you asking this question?
perfect, thank you.
Yeah, like this data and these points were done by like a postdoc in our lab, so it was like that project that I was kind of thinking back onto for mine. But anything in the field, like this kind of structure worked for my project because we had a model that we were working on, had specific findings that led to my aims. Where that's the information that you want to include is like what kind of system are you working in and what kind of preliminary data directly ties into published stuff, anything in the field that directly ties into your project.
I'd say it's also good here, you notice he didn't take up the whole board.
Yeah.
Yeah, cause like, you will likely get a lot of questions in the beginning. And off of your talk, I didn't, but like, I'm told that's what happens. Yeah, like all of mine like went straight into Gigabait and I was like, cool. So like make sure you're leaving blank space and like, you see his drawings are intentionally big, so if someone asked him to fill in, a pathway or whatever, he could do that. So think about that when you're, practicing. I also recommend like, go to a whiteboard and practice this, don't just practice on a piece of paper. 4th floor of Light Hall, there's all these classrooms that are always unlocked and open and
All
markers.
those, like there's like study books in the medical library, we just would post up there for hours and take over those whiteboards, practice with everything.
And we'll practice this too on our last week.
How many, people have gotten started thinking about their chalk talk or putting something together? Yep, very good. Thinking, yes.
Yeah.
Do you all think you'll have opportunities to practice it with your lab? Because your lab mates, your PI are going to be the people that know your project as much in depth as you do, if not more. So like they'd be able to provide more constructive feedback.
So did a lot of you coming through IGP and GP mix?
So the PIs can provide feedback?
Yes.
On the-
They can,
Like
okay.
Is that
this,
been-
yes,
I
for
think
the
like your
review,
other
no.
Huh?
For the chalk talk, yes, but not for the review.
Oh, okay.
Yeah.
Neuro?
So there, you can
We'll
ask.
have them go through stuff as well.
Should definitely ask them.
Just for like giving good information. And so right away, some of the questions that I got from this were about neuroproliferation, and so Rich similarly had me draw out the corticogenesis from like neural tube formation on. Which like I was prepared for that question, so I was all good, but like that was right away they just jumped into like, hey, describe how the brain is built. And then from there, it's like, okay, so then what role did microglia have in it? So like the nature of questions that I think are fair to expect are any basic kind of textbook information aspect of your background of your project, fair game, and then where you can like do a really good job with your oral part of the exam is explaining how a specific question or your area of interest ties into that textbook. So I was able to explain like how microglia are involved in the corticogenesis process at each kind of each step with it, and after the fact I was getting feedback.
Go, go, go.
Trying to like that had saved me from passing my qual a little bit. My written section was not good. Like they said it was bad enough, like they were considering it being like a conditional pass, but then I did good enough on this part of it where they were like, okay, he's cool. So you can always make up for it. The written part feels weak, you can always make up for it with your actual questioning and everything.
who wants to volunteer to do the two drawings today? The tripartite maps and then myelination. Not Raleigh or Will because they do most of the drawings, so it has to be someone else. You can bring your notes with you for this, okay? Like, I'm not gonna. If someone can use this whiteboard too, if you want to go at the same time, like, pick one.
Can I just focus on the malnutrition?
Foundation fellowship.
Yeah, you can pick, if you want to pick myelination, and someone else can draw
It's
the
very
tripartite
unfortunate
map.
because all he applied during undergrad and the review he got back was
So
very
these-
good.
I'll be talking about myelination today and the key differences in the central nervous system and the peripheral nervous system. And so we're gonna start.
Like last year, anyway.
Okay.
I stopped it from
So the
25.
two main differences between the central nervous system and the peripheral nervous system when it comes to myelination are the cell types. And so in the central nervous system, we have the oligodendrocyte.
They did a better job of telling you about fellowships. Here they don't tell you anything.
And in the peripheral nervous system, we have the Schwann cells. And so what is really important between these two different cell types is that the oligodendrocytes are made from the neuroectoderm, whereas the Schwann cells are made from the neuronal crest. And so these two cell types make up the myelination. I'm going to draw an axon. So don't judge my drawing. Okay. So here we have the axon terminals, with the cell body, the dendrocytes, which are the cell processes. And I'm going to use a different color. And what actually wraps around in concentric circles, and I believe I read a 2018 paper where the myelin sheath actually wraps around the axon around 30 times, more or less. So it forms this myelin sheath, and as you see, I made a little gap here because when an action potential is generated and comes through this axon, it will actually be able to jump. And that is called cellulatory conduction. This is hard to do. Yes, yes, thank you. Conduction. Saltatory. Yeah, saltatory conduction. And this gap is called the node of, or nodes of Ranvier.
Don't look at me, I was saying that the last one wrong.
So would this be a good place to stop, or should I keep going? Like, ask for a line of questioning.
They may interrupt you like while you're doing that, especially if you start by being like neuroectoderm, neurocrest, like that'll get you right into neurodevelopment.
Right.
And I know
My
we're at
question
the
is-
generation lab, so like, yeah, so they'll probably ask you like right at the beginning, specifics before you
But
like
be
get to
careful.
drawing it.
So should I slow down? Like a nod?
Yeah, I think
Yeah,
what's
I felt like I was
tough
going really
is-
quickly.
I thought your pace was fine. I think what's tough is, like, in the qual, you'll be asked a direct question
Right.
about it, where this is kind of like an open-ended prompt of like, hey, draw myelination in the CNS and VNS. And so for answering questions, you always want to make sure that you are answering the question, and then let them ask more.
Okay.
So where you get into sometimes like different rabbit holes or opening yourself up for neurodevelopment stuff, is you're providing thorough information, you're providing complete information. You're doing it correctly, right? Answering all of the question as you see fit, but you're also opening up a bunch of new avenues. So like right away, it's, right, why does the derivation from neuroectoderm versus neurocrest matter? How do you generate an action potential? Is it, like, are all axons myelinated at the same degree? Like, is it, how do they, how does the myelination actually differ between CNS and PNS, like those sorts of things, just right off the bat.
And if you bring up like a jump,
Mhm.
I know like you could get asked about like what's specifically happening with the ion channels, like what, like the super concentrated in the
sodium
nodes, right?
channels,
Yeah.
right.
So, like Matt said, like conduction of an action potential, like they'll get into the specifics in like the ion level and like why is that jump important and why, like what is the myelin sheath made up of? You know,
Yeah, and what happens?
The white matter, or it's like a white,
Right,
yes, it's a fatty substance.
yeah, lipids.
The white matter is the region you call the brain, but it is fat, lipids.
for sure. And someone might ask you a question like, okay, why is, since you brought up kind of like the nodes of grandia and action potentials, why are myelin important for action potentials? And what is there a disease where myelin is degenerated?
Yeah, so myelin is really important because it acts like as an insulator, so it acts to speed up the propagation of the action potentials to allow the ions to flow quicker down the axon. And a prominent disease that is known is multiple sclerosis as it degenerates with malnation.
I believe ALS.
And I would just stick to one, and then maybe I'll say another. Okay. But yeah, I need to brush up more on this, my neuro development, so.
If they ask about all of those, don't like wait for them to specifically ask like, okay, where does it come from?
Okay.
Development, like I wouldn't just like let
So
that
start
out
with
because
this.
then they'll be like, okay, we're going to development.
Okay.
Yeah, like if they ask about all rodenticides versus Schwann cells, be like, one's central nervous system, one's peripheral nervous system. And that's like an answer to their question, right? And then they'll probably ask you like what does it do, right? And then you can talk about the myelin and you'll get into, I'm sure, like, if you bring in the development stuff, they will dive into development, you know, like they'll
Yeah, especially.
use what you have on the board.
No, I suppose the true difference, like separation that happens is the neural seed from the neural cells, right? They are both from the neural germ layer, so.
And then there's a, what is the difference? Like how do they, why are they different? Or
Like their actual function.
like they're different because they're in the CNS, within the PNS, but how do they?
How do they actually differ?
I feel like it's really obvious. Does anyone else know?
Oligodendrocytes myelinate multiple axons. Myelinate one.
So it goes in how the processes that will attach to multiple axons and then have them, but also is a cell on the axon that will build and wrap.
So I actually learned this as a really funny acronym, so like your dendrocytes are like axons. They have many arms.
Oh, I like that.
Schwann cells is more like a snake, it's wrapped around one. So there you go.
Then if you end up with on your qual committee, he will probably ask about that, because his whole MO is peripheral nervous system development.
Okay, sounds like we're gonna try some.
Does that someone want to talk about gray matter and white matter regions of the brain first before we move on? You don't have to draw if you don't want to. But if you just key regions, what's the cortex? Is that gray matter or is it white matter?
Great.
These are things they'll ask you. And like, they made me draw both a human and a mouse brain. I don't know why, but to show. So, no brain regions. You have to know all of them, but like.
I was asked to draw the brain's label 5 regions as like a throwaway question to the mind, and I couldn't do it, because 5 points was like so brain dead and it was so simple a question that I hadn't really thought about it. And so I kind of walked on it, but we're still okay. But
Fiona likes to ask really like if we've ever met Fiona. Yeah, maybe she'll learn and be like, okay, draw brain, and then what's your favorite part? and like she'll keep it that general, and so just like don't panic if you get something like that. It is that simple. Like you can pick your favorite, or like the one you know the most about.
Yeah.
If you have an open-ended question like that, and pick something that you like
Huh?
really well.
Which one do you like?
Alright, tripartite synapse. You all, we've done it before. You guys have got to get up on the board though.
Yeah.
It's different than just talking.
It is, you forget everything when you get up there.
Yeah. No, I'm concerned for anyone who hasn't gone up
You
yet.
can
to be
bring your
honest.
notes with you if you want to. Like, obviously you can't in the fall, but today, if you just want to practice drawing, bring your notes, it's fine. But like, someone can do it. And people can help fill it in. Like, if you want to pick a pathway, you can make you want to. People can do it. But just someone get up and start drawing. There's a whole basket of markers you can try for these. Cool. Alright, let's start filling it in.
What are we looking at?
Mimi.
What kind of synapse is it?
So this is a tripartite synapse, and the pre-synapse is gonna release glutamate, so these are gonna be glutamatergic.
Fill it in. You can help people.
Do you want me to fill in, like, start going
Sure.
neurotransmitter? Is
Yeah.
that what-
Sure.
Can you like describe it out loud while you're going through? Like describe what you're drawing.
Okay.
It's good to practice like talking through while you're drawing, yeah.
So in the pre-synapse, glutamate is, they make it from glucose. So glucose comes in, and through the glycolysis to acetylCoA is transferred in the mitochondria and through the T cells glutamate from glutarate, which is, this is glutamate decarboxylase, I believe. Yes. Okay, then also in the, well, we'll just keep and then glutamate can be released in vesicles. Transported in through the circular transporter. Killing it.
You're doing good.
Thank you. And the circular transporter is a proton on ATPase. And then will be released into the synaptic cleft. Where combine, and then the post-synapse, you have metabotropic glutamate receptors. There's also NMDA, AMPA. And then it can be uptake into the astrocyte via the glass transporter, also called excited transporter. This is glutamate aspartate transporter. Here's broken down into glutamine, which is glutamine synthase. Pardon?
Synthetase.
Synthetase, thank you. And then glutamine can be transported out. This is SN.
Carrier.
What is it? SN1? Where then can be reuptake also into the presynapse and converted back to glutamate. I think this is also an EAT.
So what goes, convert it back into glutamate, or what first?
I have glutamine.
Wait, glutamine?
Yeah, I have glutamine up here, which I put in,
Yes.
and can then go back into glutamate.
And what's the enzyme to do that between glutamine, what converts the glutamine to glutamine?
Glutaminase?
Yes.
Is anybody's project gonna be on? Release.
We covered some of that with Shannon's net manipulating too, so.
Do we want to get into receptors at all, or?
We've talked about it, do you guys wanna talk, we talked about it a little bit with Elena and Shane and Zara.
Because that was excellent explanation, and then questions that they'll, because that's, at least when I was preparing for the call, that was the thing that was the most difficult, was trying to expect what kind of questions you get, as well as setting yourself up to get good questions. And so, all this information is correct and great, but then immediately be like, okay, so how do each of those postsynaptic receptors work? Be prepared for that. But the information you're providing, you kind of have to be ready to answer at least one or two follow-up questions on.
And they can also ask like questions if you have if you don't have room experience. What's that called? Cytotoxicity.
And if you want to get really fancy with it, since it's typically a day, what else is it, what's missing from the tripartite synapse?
Microglia.
Microglia, yeah. So is it, that's the quadpartite synapse, where microglia is actually ATP, that's released as part of this. That's involved in spine regulation.
Pruning.
Pruning, yeah.
Would it be ramified, or would you just say microglia?
You can just say microglia.
Okay.
There's a really cool recent paper that actually shows coordination between astrocytes and microglia in synaptic pruning. So astrocytes will retract from the tripartite synapse and allow microglia to get in there and clear away. So that's pruning that way. That's just a.
And then they'd be like, what's pruning?
Yeah. And then they get the whole other thyroid that way.
So astrocytes have a number of really important functions at the synapse and with neuron, metabolic processes. Does anyone want to talk about some of those?
Where else are the astrocytic end feed?
Blood brain.
So,
It's wrapped around the arteries, or wrapped around the blood vessels, or on the basal
so
membrane.
she's asking about metabolic response.
Right, like immune function, regulation, it maintains the neuronal, or helps maintain the neuronal barrier. I'm trying to think of overall function. So maintains, regulates, and protects.
So
That's how I
if it's
think.
on the blood-brain barrier, so in the synapse.
What do you get from the blood? What does the brain get from blood?
Oxygen.
Oxygen?
And glucose.
Glucose, right. What do neurons need a lot of?
Not the blue color maybe?
So you can think about it as sort of shuttling nutrients and stuff directly from the blood-brain barrier to synapses.
They can also like sense when like there's signaling. So when you have like high metabolic demand, because two neurons are signaling, the astrocyte can pick it up, and often it's calcium. Some calcium within the astrocyte, and that can signal for vasodilation. You don't have to go into like the details of it, like my project would, but like I don't think. But know that like astrocytes are important in like that regulation and getting the delivery of like glucose and oxygen to the right places. And it's because this type symptoms.
We'll talk a lot more about astrocytes too when we talk about development next week. So get kind of prepared for more. You also hear about it. So.
It could even, like for folks who might not be cellular molecular level, like this kind of stuff is then what would create a inhibitory tone or inhibitory tone that I'm looking at though, because then I does this kind of system cell stuff that would get picked up on fMRI signals and on that. These are the cellular mediators of the signal that you're detecting for wider, larger scale changes. And so a question that you might get in this setting is if the EAT is gone or dysfunctional, what would happen?
The EAT is not. Okay, so that means the glutamine cannot be re-up. So you can't reuptake into the pre-synapse and create more glutamate.
Yep.
So would that be less? Would be a reduction in the amount of glutamate,
Yeah.
excitatory signal that's produced.
That question, right, following along with the logic that you have there, and that's kind of what they're looking for is when you're asked questions that you might not know, they're still able to look at what you've drawn or draw upon what you do know and get to an answer, get to some kind of answer. So that's good.
This also goes into what we talk about with LTP and LTD. So astrocytes and microglia are really important in strengthening or dampening in the synapse.
Mentioned it a little bit, but that one of the other things that happens with glutamate release of ATP sometimes. And so that now we have another molecule in here that could recruit another cell type to this area. What's that molecule? I know it's called ATP, but like what genre molecule would that be? Molecule that recruits other cells to it. Yep. Maybe I pushed it away. That would be huge next week for development stuff. It's all interesting. Robo signaling, all of that stuff that I know we don't know right now.
So in this case, ATP acts as a chemo attractant
Yeah.
to microglia.
Yeah.
Cool.
It's also, ATP can be released in cell death too. So like the general term is chemokine signaling, either attract or repel. But ATP is a common molecule during neuronal cell death to attract some sort of cellular response either and from outside the microglia. Jump into inflammation.
Yeah,
Yeah.
so what else to astrocytes really involved in? What do they make?
Inflammation.
How do they respond to inflammation?
Astrocytes can produce interleukin prostaglandins. They're derivatives of arachidonians.
Nice. What is that category of molecules called?
Cytokines.
Yep. Yeah, cytokines, chemokines. I think of chemokines. I think of chemoattractants, chemorepellents. So that's why it's like signaling molecules to move cells around. And then cytokines, I just think of like cytocell inflammation. That's just a bridge that I have in mind for how to tell those apart. But is inflammation always bad? when is inflammation good?
And healing.
Healing, right? Pain,
Fighting infection.
fighting infections, right? So what is it doing in those circumstances that's good?
Engaging in cells, blood flow.
Yeah, so it's the help signal, right? It's getting the immune cells to the area that needs help. Which immune cells in the brain? What are they?
Bye-bye.
You learn anything today, so I like
No,
that.
you do.
But no, like that, right, it's not always only microglia as the immune cells in the brain. What are some instances where you get immune cells, other
Leaky blood-brain barrier.
leaky blood-brain barrier?
You have immune cells within your brain.
Very vascular space, very vascular macrophages, absolutely. Okay, Grant comments on that. Any other cases you can think of? Really bad cases? Big old puncture wound to the brain, any kind of traumatic brain injury, any kind of straight up rupturing of the blood-brain barrier. You can have infiltration and leakage of cells in the brain. And where I think that that's especially bad is that you then have professional, sort of like professional immune cells that are really good at ramping up inflammation. And the brain is not very good at handling a lot of inflammation. And so that's why it's so sensitive, is because it's sort of like a specialized organ tissue that's not used to typical peripheral immune cells. So when they are then in the brain parenchymal space, not good. It's just, the cells are too strong for your neurons to handle. They aren't very metabolically sensitive, so it just leads to damaging signaling much more quickly.
So everyone here is either studying some sort of neurological disease, disorder. Does everyone have an understanding in their own system what's going on with inflammatory responses? Every single one of your guys' systems, there's changes in glia and inflammatory signaling. I would have a general understanding of what those are. Some is actually glia size, some is glia number. Where regional specificity, I would know what those are generally for your specific system. So you probably, if you're like doing, schizophrenia research, you're probably not going to get a bunch of questions with, reactive astrocytes and gliosis in Alzheimer's disease. But it's fair game for you to know what's going on in schizophrenia in that instance. So know what those are.
Yeah, well, I saw a little shrug there.
Yeah, I'm trying to, I don't know.
Because I guess, yeah, that lends itself to a good question of these glial cells that we're talking about today, how they respond to neuroinflammation, right? So that microglia, and because that's what I'm gonna harp on, but like microglia, for example, they usually are meant to kind of maintain homeostasis, but just as basic as you can think of, what happens in microglia when inflammation is high? What are they doing? Do you have like activated microglia?
Going to the area.
Yeah, they're gonna go to that area, so you're gonna have sort of swarm or just microglia there in the area, so cell count in there is one way to measure it, but that microglia also engulf stuff a lot. And so hyperactive microglia will be clearing away too many synapses or too many neurons. To a point where it could be then, people think it's one of the underlying causes of atrophy in Alzheimer's disease, is that you have hyperactive microglia that are clearing away too many neurons. The astrocytes are hyperinflamed. Not as certain on this one, but what happens?
You'll hear all about it tomorrow in my talk.
Perfect.
No, reactive astrocytes are really key, but they're also really important for the recruitment of, for example, microglia, and they're thought to be the first responders in a lot of diseases and neurological disorders, to drive their number changes in everything from autism to Parkinson's to schizophrenia to any sort of dementia. There's changes in glia usually first, and so I would know what's relevant to your system. Generally speaking, like what's relevant to whatever you're doing. Even if you're looking at neurons are not alone. So you need to think about what else is happening And especially if there's, if you have a mouse model, which we'll talk about more probably next week, since we're getting low in time. If you have a mouse model for disease, knowing differences between human and mouse, both neurons and cells, there's size and number differences. Does anyone know, are neurons bigger or smaller in human versus mouse? Those work with mice.
Human neurons are bigger.
Actually,
Smaller.
there's more of them, but they're
More.
smaller. Yeah. Yeah, it is weird.
There's another thing that astrocytes do, right? That's like kind of on the list. Involved in antioxidant response. Can people give like examples of antioxidants? C. Vitamin C. We love that one.
I would say for this, like, so a lot of the questions that I pulled for your review questions, are from Fiona's fundamentals lecture, and you guys have the recording, so I would definitely watch that, like, just to have an overview. She goes through the chemistry very well, so like, just rewatch that, revisit it, know what all those terms are, know a couple examples of them, how they relate to your disease specifically. Those are the ones that are very, they're not probably not gonna ask something completely random off of something you're not involved in with this.
Especially for the folks who are cellular molecular, try and just relate anything to what your project is at the cellular level. It's hard.
I will say it doesn't always stick to the project. I'm very much off the project. Like I got like a couple of cerebrovascular questions, and then the rest was like I got into endosomes, and I haven't studied that since IGP. So it's not always, but so, like you definitely try and relate it to your model or like whatever you're studying, but don't limit yourself and like limit your studying to like just around your project, because it's not gonna like stay there necessarily. Some people do get exams like that, it's nice, but it's not always the case.
Last question without any clear stuff.
Does anyone have a disease model they're working with? Those are especially fair game. Does anyone work with mice? I know there are people who won't. Yes, okay. I was warned by faculty that, and I quote, I better know every damn thing about my model and be able to prove to them why it's the best model. So you need to know everything about the genetics, how it's made, the background, differences in mouse background. If you have mixed backgrounds, what that means for your model, how that means for your experimental design. You design experiments differently if the mouse model is congenic versus isogenic versus co-isogenic. All of these things are, if you work with mice, all of these things are fair game. We'll go to
Like the background, the genotype of, is it,
Like
if you're
how
not
to-
a part of,
I mean, like how to genotype or just like?
yeah, like if you work with mice models.
You should definitely, if you're working with a transgenic model, you should know everything about how that model is made.
Okay, I just started working with mice, so
Yeah.
I'm getting into it, but I'm just.
Yeah, like I would know everything from like blastocysts, like that's a very common like development question too, because they can use your mouse model to go through development. So I would know everything from blastocysts to if there was a cassette they used to make the notch. We'll talk about gene editing next week. I would know everything, and you need to be able to prove that your mouse model is the best one for your system. If you don't work with mice, I know several people who say those people who don't work with mice, they love to use the low-hanging like Cree, just to show that you know something. So I would please memorize that, know what Cree is, know what Cree ER is, the differences in what you can do with them. I would also know general overview of different models for your disease. So I work with the 5XFAE, there's also the one Alzheimer's mouse, there's the APOE model mice. I would know like 3 or 4, and why yours is the best for what you are studying. What are the caveats to using that mouse? What are the advantages of using that mouse? I was asked a question if I was going to design the perfect mouse, which I'm trying to do right now, like what that would look like, why I'm choosing that promoter, why I'm using those genes, how I'm going to use those genes, how I'm going to validate it, and these are all like experimental questions that if you work with mice, they'll ask you. If you don't work with mice, they might still ask you to design an experiment around an animal model. So I would know at least a couple that are common for your system.
Yeah, orthogonal or alternative approaches will be really important. A lot of my questions were just about, okay, what would you do if you're not gonna do RNA-seq, give me something else. So, and if you are proposing a certain like experimental approach, you know it from beginning to end, even if you ship off part of it to your collaborator or to a core on campus. So I can't just be like, oh, and here are some, I'm gonna do TEM imaging. Okay, well, how do you prep the sample? How do you do the data analysis? Those are fair game too. So really, really know your methods and your models.
And common ones, even if you're not using them, common ones that other people use for similar things you're studying. And you need to be able to talk about why you're not doing that and you're doing whatever you're doing instead. That's really important.
It just has to fit your question. That's it. Like, it doesn't have to be the top of the line, best, newest thing. It just has to make sense.
That's what I was gonna get to, is like some of the explanations for it, like you should definitely have a valid scientific argument for why you're using your model or other models. Cost, reproducibility, popularity within the field, those are also valid reasons to use a certain model. So like, while it's not the best science answer to give, it is a good answer to give as well, because it's a practicality thing. It's a practicality limitation of like, why aren't you using this model? It's like, well, that model is only orderable, like you can only order through Jack, it's $18,000 for one mouse, and so it's not practical for our lab to spend that much money for one mouse, right?
And
So
that's feasible.
that's
As long
exaggeration,
as you
but
know that, yes, there is another way, but here's why we're choosing this one,
Yeah.
right?
And I'm saying, if you come across any specific questions to your stuff, we've got our emails. Please don't hesitate to ask.
We'll talk about them more during development too, because they're really important for that. Neuronal migration, those, okay.
If you have any questions on Fiona lecture too, you can
Have y'all started putting in your committee members in the spreadsheet? Setting up meetings with your committee members? I'm gonna repeat that at the end of every session. I got a couple more, but meet with your committee members, practice your talk on the board. It will take you very far.